So imagine you walk into a room, and that room, based on your previous experience with it, changes the way that your immune system functions.
I just wanted to be like, gives you heroin.
Gives you heroin. Wouldn't that be nice?
You walk into this room and you get heroin.
Yes.
Hi everyone, Susanna Harris here, and I am excited to host the Fresh Press Science episode 5. And usually Gabi, Serrato, Marks, and I will get together and talk about an exciting new topic that's in the media or that's spreading around the internet like crazy. And we talk about what this topic is, and what's really interesting is that we always try to find a scientific author of an actual journal article who can come on and give us some expert insight. And this week, I am super excited because the topic is going to be the placebo effect and what that really is and why we should care about it. And unfortunately, Gabi is not here. She's on vacation, which awesome. Hello, self-care. But what's more exciting is that I have convinced someone that I know to come on and talk about her exciting research paper. So if everyone would please say hello to my friend Christina.
Oh, hi!
Christina and I know each other because both of us are graduate students here at UNC. And do you want to tell them a little bit about yourself? And
Oh gosh, me. So I was actually originally from North Carolina, so going to UNC was not a big jump for me.
it's from Iowa.
Nice. Coming from Iowa. I actually might be going there. You'll have to give me some tips.
Yay, good for you.
Let's see, what about me? I'm a gamer, I'm a nerd. I am all the stereotypical science plus nerdisms.
You have awesome science jewelry.
Do I?
Yeah.
Yes.
Yeah, you always have like some... What do you have?
I have neurons. I've got some Harry Potter horse or the Deathly Hallows.
Yeah.
I have ridiculous things. I have a time turner. I've got the Evan Star from Lord of the Rings.
Okay, that's what I thought you had. And I was like, if this is not right, it's going to be so
it's just, it was hard to narrow it down.
The science that we're going to talk about today that Christina does is neurobiology, psychology, looking at what's called the placebo effect. And hopefully some of you watching have heard of the placebo effect. If you've heard about it, if you're interested in talking about it, feel free to leave a comment. You were saying that these mice would bite, but rats...
Rats,
good.
yeah.
But also, are the rats good because you're giving them heroin?
Okay, that helps, for sure. But most of the time, we're not giving them heroin. Let me just clarify.
That was so many of my questions.
What do they like when they're high?
I mean, is it hard to get?
Like all the abuse drugs, it is very tightly regulated by the government. So you do have to have a DEA license.
Oh.
They do inspections. It's a big deal.
If I was just going to quickly say this study, it would be like, you give rats heroin and make them really sick.
Yeah. No,
that's it. So why do you like this?
Well, they weren't hiring at Denny's. And I was like, here's my skill set and it happened to be research with rats. Yeah, so I actually ended up, I came from a biology background, so I was an evolutionary biologist, and I was, funding cycles. I got laid off. So what happened was I was interviewing for jobs, and I read this study from this lab, and I was like, oh, I gotta know how that works.
Yeah.
So that's how it all started, was with me just geeking out about this project, honestly.
We met through outreach at the University of North Carolina, specifically through the Moorhead Planetarium. So good. Shout out to Tamara Pohl.
Oh, Tamara!
She's amazing.
She's my twin.
Has
she told you that she's my twin?
I've seen it in action. I have seen it.
It's terrifying.
I told her we're going to send her this video once it's edited. So.
Oh, no. I'm going to hear from her.
So you wrote this. What did you write this for?
So I wrote that for the original ComSci Con that I attended before I started organizing it. So
you're such a badass.
This was the first one. I think it was in 2014. Something around there. And this is the one that got me involved in all of the ComSci Con goodness. I love it. Shout out. ComSci-Con is amazing.
The article is called When Learning is Infectious, A Placebo Effect Beyond Belief, which is not an overextension here.
I always do play on words.
But it's great. So, quickly to paraphrase, it was this study in the 70s that was looking at, so they would suppress their immune systems they would suppress these rats' immune systems while they give them sugary water.
Well, so what's interesting is that that's not what they were shooting for at all. It turns out that what they were trying to do was study Have you ever had food poisoning? Do you eat the food that you got sick off of? Exactly. I mean, this is across species. Rats hate it. People hate it if they've gotten sick. And that's evolutionarily adaptive, right? Because you don't want to eat something that's going to make you sick. And you don't have much time to learn that connection. Just like that happens in rats. And that's what they were actually studying. It's called conditioned taste aversion, which just means trained aversion to a certain taste. Once they started training them with this drug, they were getting sick and dying when they just drank the sugar water. So when they were testing to see how much of that food that made them sick they wanted to drink, they were like, they would drink, they drink a little bit of it and then they would die. But it was just sugar water, and they weren't getting that nauseous drug anymore. So what was happening? So it was this connection between this drug that makes them sick and the sugar water that even afterwards was having effects when they just drink the sugar water. Robert Ader, who's the person who did the study with Nicholas Cohen, and he said, no, I'm going to figure out what happened. And so very systematically they tested, and it turns out that their immune systems were being suppressed by this drug. They didn't know that was a side effect of this drug, that it would suppress the immune system.
So the drug they were giving with the sugar water.
Exactly. The one to make them nauseous. So what happened was afterwards, when they were just drinking the sugar water, the body recalled that immune suppression that happens with the drug because they were connected by learning.
When I think of placebo effect, right, I'm biased. I'm always thinking of those. I always think of those people who are like, you're gonna hold the purple crystal.
You were gonna hold the crystal, and it's gonna make us feel whatever purple inside.
Yeah, but obviously these rats weren't like hippies.
Right.
The placebo effects that these rats were experiencing, that they drink sugar water, their body said, okay, when we drink sugar water, our immune system is suppressed. Then, so their body, whenever it drank sugar water, would have a suppressed immune system. Was actually killing the rats.
Yes. There was an infection that was spreading around the colony, and they were more susceptible to it because they were immune suppressed.
That is amazing.
This tells you a really important thing about placebo effect, is that, it seems like it's voodoo crystal, but really it's the result of learning. And it may not be conscious learning either. So it seems that the brain has an innate way to make connections between things to allow it to make predictions about what is going to happen or what it should do.
Makes sense.
Yeah. So really all the placebo effect is learning about connections between things. The drug was immunosuppressive. So immune suppression is what got connected. Opioids are also immunosuppressive. But if the drug was a pain reliever, it would do the same thing. So pain relief has actually been shown to be very, very subject to the placebo effect. And for humans, it's a little bit more complicated because we use more information, or we use information that we're more aware of, like social cues. How competent does this doctor seem? What is the doctor wearing? What kind of biases may I have against the doctor's sex or race or anything? Those affect how you respond to treatment.
Nocebo.
Nocebo! Yeah, that's my people. Nocebo is the reverse of a placebo. So where it's where your beliefs can cause you to actually block the effects of an active drug.
Wow.
Right? So now we're not talking about you believing something and it happening. We're talking about you not believing in something and therefore it doesn't happen.
Wow.
With real drugs that are doing pharmacological things.
It's stuff like that where I'm like, we don't know so much.
Oh my gosh, especially when it comes to the brain. This isn't just with pain and immune suppression. This is with a lot of different things. So Parkinson's patients, they're actually have a depletion of dopamine in their brain. We think of it as a pleasure drug. It does all these other things, but I'm not going to be that person. So the dopamine is actually depleted in their motor areas. And so one treatment is to give them the precursor to make dopamine. It's not a great drug to be on long term, and half of it was sugar pills, half of it was the actual active drug, and it turns out they did brain scans. They were looking at the amount of dopamine in the brain, and when you told them that they were going to get dopamine, they got more dopamine in the areas that the drug usually provides dopamine to. Right?
Should we just throw away all medications? Does it matter?
Well, so here's the thing. I mean, the way that we design drugs is that we compare them to placebo. That's what drugs are, and sometimes it's a very small amount above what placebo can do. If you can modulate the effect of an active drug,
sure,
that might be one reason that a lot of drugs may not work for certain people.
Sure. Preconceived notions about what is going to work. That could
right.
Totally affect
and
it.
How we deliver those and how we treat them and the information that people get about drugs can affect everything about that drug working for that person. I think more so what we need to do is we need to figure out, once we know that a drug is effective above placebo, on average, we need to figure out how to get the maximum effect of that drug from the most amount of people. By harnessing the placebo, by enhancing the way that we deliver it. What does the pill look like? And that's going to change with everybody's experience. Because the placebo effect is extremely variable. How do you control people's beliefs? You know, like, if we knew how to do that, I feel like there would be a lot less political strife. We'd all get along. We'd all come to reasonable agreements. It would be amazing. Changing people's belief systems is really hard. So one way that you can do that is through intentional training, right? So to expose someone to associations that actually guide their placebo effect. But furthermore, and the reason that I like the immune suppression so much is because it seems to, in at least one study, be less affected by patient expectations than it is from the stimuli that are connected with the active drug.
Okay, so I'm just going to hold this up to the screen.
Just to say, oh God.
So, that's,
Yeah. We keep trying to come up with an acronym, but there's so many other acronyms in this paper.
Well, so here's the thing: is I read the stuff she sent before, which is like, oh, cool! Like, this is a cool effect. I can get down with this.
Yeah.
Like your Psychom was great. Thank you. And then I got to the paper and I was like, thank goodness for the Psychom, because there is no way.
No, and that's what's really frustrating, because this is, oh God, we are not that many people study what I'm studying. So there's a group in Germany that have done this with people. So we know that this conditioned immune suppression, this is what I call it in the paper, I'm so sorry. It basically means you've trained your immune system to respond to a thing that it wouldn't normally respond to if you didn't have that training. Yeah. There you go. And in this case, we're connecting it with a context. So a place. So imagine you walk into a room, and that room, based on your previous experience with it, changes the way that your immune system functions.
I just wanted to be like, gives you heroin.
Gives you heroin. Wouldn't that be nice?
You walk into this room and you get heroin.
Yes.
The title of this paper is Acquisition of Heroin-Conditioned Immunosuppression Requires IL-1 Signaling in the Dorsal Hippocampus. So,
is there any word in there that's not jargon?
I feel like acquisition, like, I think our general audience gets that. So, and heroin is an opiate, right? So what does heroin do? Why do people take heroin?
Oh, it feels good. Like, people compare things that they enjoy to heroin when it's like, oh, that's like heroin. Like Al's Burger Chuck, which I had today, which was amazing.
Yeah, I'm not sure this is an advertisement.
No,
for it.
drugs will ruin your effing life, okay? So don't do drugs. Yeah. We study this.
Have burgers. Go to Al's.
Have burgers and then compare it to heroin.
Come to UNC, come to Chapel Hill, come hang out with us.
Oh my God, Al's Burgers.
And we'll go to Al's Burgers.
Oh, so good. No one gets prescribed heroin, which is the good news, because it is so addictive. But people do get prescribed morphine, and we've done these same studies with morphine too. Okay. Yeah.
So immunosuppression. Let's talk a little bit about how you measured immunosuppression.
That's an evil story. So the way that we measure immune suppression, we can't actually make animals sick. With an infection, that would require a lot of additional approvals, and it would be really kind of mean to go, which ones died? You can't do that in an animal research study. So what we do is we challenge their immune system, and the way that we do that is we take a piece of bacterial walls. Your body will recognize it as if you are being infected by this bacteria, but you're not. So it allows us to use it safely in the lab. If we were to inject ourselves with it, we would also get sick. But basically it stimulates the immune system. And so what we can do is measure the amount of stimulation of the immune system that occurs to this injection of LPS.
Okay, so immunosuppression, we've turned down that immune response.
Yeah.
We're going to skip IL-1 just for a second, but then you say...
Please.
Yeah, you say dorsal hippocampus.
Mm-hmm.
Okay, so when I started reading this paper, I was like, oh, cool, we're going to learn about the immune system. And I started reading, I was like, okay, not really. We're actually learning about the brain?
Mm-hmm.
Okay.
So, it turns out when people say the placebo effect is all in your mind, they're kind of right, but what that doesn't mean is that it's imaginary.
So your brain is controlling the response.
Right, exactly.
Huh.
So we can
it's not
actually...
like your lymph nodes?
You would think it would be some sort of immune organ, but it turns out the brain is actually responsible for controlling a lot of concerted peripheral effects.
Wow.
So it can change your heart rate, right? Because you've got to be able to sense danger, and you do that with your eyes, which is processed by your brain, and then it sends the thing. So your brain actually does all of the mediating between your external environment and what your body needs to do. Mm-hmm. So, yeah, we found that you can actually block a certain gene in a certain place, and that prevents the ability of that place to suppress the immune system after training.
Okay, so we got acquisition of heroin conditioned immunosuppression. Heroin turns off your immune system, requires some signaling in the dorsal hippocampus, in part of your brain that does stuff.
It does stuff.
I took psychology 8 years ago now.
I had one psych class before grad school.
That is amazing.
Imposter syndrome.
Imposter syndrome. Okay, so this was a study on 55 mice. And I fed up. Rats.
Only a rat person is going to say that, or maybe it might
people on here will say that.
Okay.
I'm going to get hate mail from people I'm going to get some side-by-side description of what a rat is versus a mouse. I would love that. Feel free to post that, put in your stories, tag us. Hi, buddy. This is neither a mouse nor a rat. This is a dog.
The hippocampus is really responsible for episodic memory. So if I remember, I went to Al's Burger Shack today. Yeah, that was good. That's episodic memory. If I wanted to know where I was, that's the hippocampus's role as well. Or if I wanted to navigate. So it's a lot of associative stuff too. So this thing's connected to this thing, it'll do that. It'll do that with time, it'll do that with space, it'll do that with all kinds of things. So it makes connections. And it's named hippocampus for the...
Oh my gosh,
what's happening?
London taxi drivers have enlarged hippocampus.
It's true, because they have to navigate, so their hippocampal volumes are bigger. Yeah.
I'm not sure if I have one. I have no...
You definitely have one. I can almost promise you that you have one.
I just feel like I can never remember any locations, ever.
Well, it's because probably, it's not relevant necessarily to your life. No, it's not relevant. It's not relevant. Your brain only pays attention to what you need to know.
Like this. Are you checking for my hippocampus? Is it there?
You give the rats
heroin.
Yes.
That part's true.
Yes, that part is definitely true.
And then sometimes you give them this bacterial cell wall component.
We do that to everybody. So we train everyone to associate an injection of heroin with a specific place. So it's just an operant chamber. It's basically a box we let them hang out in. By the way, a high rat is kind of the cutest thing you've ever seen.
Are they just like limp?
No, they're like a little putty. They kind of ball up a little and they're like...
I'm in the wrong job.
With their eyes, and they're like, whoa. That's what they're like when you take them out. They're just just amazed by the world. So basically, we wanted to target this gene during the learning part of the pairing of the heroin with the place. To see if we could keep them from actually learning the connection so that it wasn't able to suppress their immune system. That's one way we could target it. So what I had to do was inject a drug that it targets a specific gene directly into the dorsal hippocampus. But if that...
How big is that?
It's actually pretty large. Yeah, it's pretty big. No, hippocampus is real weird. It wraps kind of around. I'm doing this it does that in humans, but it goes like this in humans instead of like this. Yeah.
Ugh. Oh.
Right. So... It's actually pretty big, and we're hitting the dorsal part of it, so that means dorsal fin as the top of the hippocampus. It's terrible. So the black dots are where we gave the actual drug that affects the gene, but the thing is, we were doing that and then training them. So we didn't want the injection procedure to be what's messing up the training, so we had to inject them on the off days when they weren't getting trained, and what we had to do there was do saline. Instead.
Okay, so and what you're trying to do here is figure out is it this IL gene?
Yes.
That's involved. So we know hippocampus and something about the immune system.
Yes, we know hippocampus, something about the immune system in this context, pun unintended context. That's what we're studying. Okay. So what we had to do was then in the other group, we would give them saline on the days they're being trained, and then the active drug on the days they weren't being trained.
Okay.
To see if it has to be active during the period they're being trained.
Did you find it was the opposite, or did I read this so wrong?
No. So it turns out that if you do it right before the training, then it has an effect. But if you do it on the intervening days when you're not being trained, the active drug, then it doesn't have an effect. So we know, I know, these graphs are horrible. So 24 hours after conditioning. So the conditioning is the training session. So this is what's so confusing about my data, is that we're actually looking for an effect. And so when our treatment works, we get no effect.
Oh.
Yes, because it's so counterintuitive, because you're usually looking for the little stars and you're like, oh, they did something. But that's our normal effect, is that it's significantly immune suppressed.
So, for instance...
That's the group where it didn't work.
Yeah. So if there is an effect...
When I'm doing talks, this is like a major part of what I need to walk through before they can go through the data, because it's just really counterintuitive.
All right. And then the second study.
Did you get the rationale behind this one?
No.
Okay. So... the interesting thing is that I could get rid of conditioning by making it so heroin's not doing anything when I'm training.
Oh.
So if I block that gene and it actually blocks heroin's effects that would normally happen, instead of the learning, well, I mean, I didn't get any conditioned immune suppression to the context. There was no immune suppression, right? So what I had to do is see if this same manipulation would mess with heroin's normal effects on the immune system.
Okay.
So that's what I did. I gave them this gene block, right, and then gave them heroin, and then gave them LPS, like right after. There's no training, there's no association. It's just one injection.
Right. So this was the part that was talking like, so blocking this gene blocks the training effect, but not heroin suppression.
Yes. So now that I know that I'm blocking learning and not the actual effects of the drug.
Do you think it's running parallel? Do you think the learning is parallel to...
I think the learning is somehow causing a concerted activity across the brain, because we know that it's not just the hippocampus. There are other brain regions too that are really important. And I think it's this communication, the circuit, when it gets activated, that causes something to happen to the peripheral immune system.
Wow. That's super cool.
We really don't understand how this works.
That's awesome. No, this was amazing. Yeah. So if anyone wants to learn more about this, look up Christina Lebonville.
I am literally, if you look up Lebonville, it's just me on the internet, which is kind of terrifying.
Whoa.
Yeah. It's nice, but I can't mess this up. I can't put anything embarrassing out there.
Lebonville and heroin.
Too late, by the way. I must search this Lebonville, and I'm like, I got you to look at my papers.
That's amazing. But are you open to people messaging you?
Oh my god, you don't even understand how much I love this. Like I said, I stuck around and went to grad school because of this project that I was getting laid off of.
Main takeaway is that placebo effect is real, even if you don't know what's happening.
Yes.
So are people who experience the placebo effect, are they faking it?
No. We can measure it. That's the thing. We can measure what's happening in the body. They're not making it up at all.
Yeah.
We can actually
the rats definitely aren't.
Yeah. We can see the brain regions that usually respond to pain not responding as much when people don't feel as much pain. And that makes you think, like, well, all the ones where we would think someone's making it up. Like, for example, you're at a party and, you know...
Oh, my God.
So, like, let's say, we didn't actually put alcohol in that punch, but people are acting stupid.
Yeah.
Is that actually causing them to feel a little drunk? Because it's connected with alcohol.
I have friends who, like, they take a sip of coffee and they're like, whew. I'm like, there's no way.
Right?
There's no way.
Yeah.
But
maybe, because our brain seems to be able to do some really amazing things.
It makes sense that if you think you're going to be in a flight or fight situation, if you think you're going to be in a painful situation, your body is going to get ready to turn on those signals.
Yes.
Because it wants to keep you safe.
It's efficient also. Like, it needs to know when things are going to happen, when to do stuff, when not to waste energy on something. And so... your thoughts are really... your brain is a prediction machine. That's all it's doing, I feel like, most of the time. And in order to do that, it has to take in all the information that it's got available. And that. Your thoughts and beliefs are one type of information that it's getting. Your beliefs are really just more information that's telling your brain, okay, this is what's going to happen. I'm pretty sure it's going to happen. Which, looking at the placebo like it's just training, right? You're just training a connection. You're training your brain to make connections between things so that it does a certain thing. Meditation could absolutely be the same way. And I thought about this a lot because I meditate. Whether I'm going to use the same guided meditation every time, will that help me get into that meditative state faster because my brain recognizes the cues and can get there sooner and be more efficient about it?
I should do that.
Honestly, I would not have gotten through grad school at all. Meditation was honestly the one thing that could shut my brain off because it's always going. It's always making predictions because it thinks the worst is going to happen, and I have to convince it it's fine.
Okay, so takeaways: don't do heroin.
Don't do heroin.
Crystals still probably crap.
Or because you believe that it will work, does it actually work?
Using this, like you said yourself, we could harness
it. Yes, why aren't we using the placebo effect? That placebo effect video is by Leah Crum, who's doing amazing work in people on expectations and how we should harness the placebo effect. Yep. And she walks through exactly what the placebo effect is, why everybody thinks it's a thing that doesn't work. It's a really great TED Talk.
Hopefully, if y'all can help us with one other thing, definitely spread the word about the Psy community because we're at like 9,600 some followers. When we get to 10,000, I know. When we get to 10,000, we can do the swipe up thing so we can get to everything so much faster. So when you're like, I want to watch that YouTube video, be there.
I don't know how YouTube works, so...
Well, it's okay.
I'm going to tell people like it because the thing happens magically.
Yeah, like the things, do the stuff. So thank you for coming on. This was so much fun. We actually need to hang out more.
Yeah, I know.
Goodbye, Tamara. Thank you for introducing us 3 years ago.
A while.
Gabi, you missed out.
I know. I was hoping to meet her.
Someday. If you have any more questions, feel free to send us a message. We'll get the edited video up as soon as possible. Feel free to ask more questions. And thank you for joining in. You're all amazing. Bye, everyone. There's so many.
You tried to go through the densest paper known to man with the dog.
Craving
your attention. Oh, I'm getting sat on.